Okay, let's dive in. We're looking at this really interesting paper today. And the big headline, kind of counterintuitive, is this. When they mapped out a lung metastasis in detail, the cancer cells with the highest long-term fitness weren't, the super aggressive ones right at the edge.
No, not at all.
Yeah.
They were actually this stable, almost epithelial-like group tucked away right next to the normal lung tissue. It's pretty fascinating.
Wild. And the reason they could even see this is because of a brand new tool, right? PEtracer.
Exactly. Prime editing lineage tracer. That's our mission today, really, to get our heads around this thing that finally lets you see cell lineage, what the cell's doing, and where it is all at once.
Which has been a huge problem, yeah. You could track the family tree of cells, the lineage.
Using things like Cas9 scarring.
Right, but then you'd completely lose the tissue map. You knew who was related, but not where they lived in the tissue.
Totally. And the other way around, you had these amazing spatial methods like MERFISH telling you exactly where cells are and what genes they're turning on.
Super high resolution.
Yeah, but zero clue about their ancestry. Who came before them? No idea.
So PEtracer bridges this gap. It uses prime editing, which is like a really precise DNA find and replace, to install these little 5-letter DNA tags, tiny marks, and they're designed to accumulate as cells divide.
So it creates this evolving barcode over time, and critically, you can read that barcode both with sequencing and with imaging. That's the key link.
And they engineered it really cleverly, didn't they? It wasn't just random mutations.
No, that's the cool part. Figure 1 really digs into this. They used what are called protospacer mismatches to actually tune how fast the prime editing happens.
So you can control the speed of the clock, like make it tick faster or slower.
Exactly. If you're looking at something happening really fast over maybe 2 or 3 weeks, you set a fast clock. Studying slower processes over months. You dial it back, it makes it super flexible.
That's incredibly useful. Okay, so they designed this tunable clock. Did they prove it works? That the imaging part is reliable?
Oh yeah, figure 2 is all about the validation. They used cells grown in a dish where they knew the lineage beforehand. Ground truth.
Smart.
And the imaging readout, the MERFISH part, it detected the barcodes really well, like over 80% detection, and the accuracy of the lineage marks themselves was super high, over 99% after filtering.
Wow, okay, so the tool is solid. They prove the imaging can reliably read the history written in the DNA.
Yep. Confirm that dual readout capability.
All right, so they've got this validated PEtracer system. Now they apply it to a real biological problem, mapping metastasis in a mouse model of breast cancer spreading to the lung.
This is where it gets really interesting, figures 5 and 6.
Okay, but before we get to the results, quick question for you listening. If you're a cancer cell trying to really succeed to set up shop long-term in a metastasis, where do you think the best spot is? Deep inside the tumor core, or right out there on the aggressive, invading edge?
Yeah, you'd probably guess the edge, right? Where all the action seems to be happening.
That's what I would think, but that's not what the PEtracer data showed, is it?
Not for long-term success, no. They reconstructed the whole 3D clonal structure and found the cancer cells basically organized themselves into 4 different groups or modules based on where they were and what genes they were expressing.
And they calculated this thing called phylogenetic fitness, basically a measure of how successful a clone was over the long haul, over weeks, based on the family tree structure.
All right. And what they found was that fitness was negatively correlated with how far the cell was from the normal lung tissue boundary.
Negatively, so the further out towards the invasive edge.
The lower the long-term fitness. Those cells at the very front, yeah, they were dividing, they were active, but it seemed transient, like scouts maybe.
Yes.
Their success wasn't really sticking, wasn't heritable in the same way.
So who are the long-term winners then?
It was this group they called Module 4, the lung-adjacent cells. These guys had the highest phylogenetic fitness. And transcriptionally, they looked more, well, epithelial, expressing markers like claudin-4, FGF1, suggesting a more stable, organized state.
Which is totally counterintuitive for metastasis, which we usually think of as this chaotic, break-everything process.
Exactly. The implication is that real, lasting success in that metastatic environment wasn't driven by just bursting out at the front. It was about finding this sweet spot, this niche, right next to the normal lung tissue. Maybe it's richer in nutrients, maybe the immune environment is different there, and adopting a stable, heritable program that thrived in that specific spot.
So the innovation here is huge. PEtracer actually fuses the lineage tracing with the high-res spatial map, in a living organism.
Yeah, it lets you ask causal questions finally, like which specific little neighborhoods, which microenvironments are actively promoting the success of these specific clones versus those ones over there.
And the potential seems massive. They mention it's scalable. You could use it to map therapy resistance.
Oh, definitely. And think about the data it generates. These huge, detailed maps combining lineage, state, and space, perfect for training new AI models to understand cell dynamics and development, stem cells, cancer, all sorts of fields.
Okay, so let's boil it down. 3 key takeaways.
1. PEtracer uses prime editing to make this tunable DNA clock, giving you cell ancestry that you can actually see spatially with imaging.
2, that combination lets you reconstruct how clones evolve in 3D, right inside the tissue, really high resolution.
And 3, the big biological surprise here, long-term success in this lung metastasis model depended on stable epithelial-like cells thriving in the niche right next to normal tissue, not the fast-growing cells at the invasive edge.
Makes you rethink metastatic strategy. Okay, one last thing to chew on. PEtracer needs some pretty heavy computation to rebuild those family trees from the DNA marks, right?
For sure. The algorithms are complex.
So given how much cells can move around in a tumor and how messy those environments are, how much uncertainty do you think might still be baked into those computational reconstructions, trying to pin down the exact ancestor in that chaos? Something to think about.
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has 3 main goals. 1, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2, to build a supportive community where we can exchange the most valuable experience-based research advice. 3, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the 3rd goal. Let's go! Let's make great science together.